Good afternoon, KUTV, Elimu Life. I'm your host today. My name is Nancy Bosibori. I'm a teacher of biology and maths. Welcome to today's session. I'm going to hand a major topic in Form 1 work, biology, called nutrition in plants and animals. Nutrition in plants and animals. So, I'm going to start by defining the term nutrition here. But before I define the term nutrition, in brief introduction, I would like to say that nutrition is one of the common characteristics in living organisms. If I may remind you, to the first topic of Form 1 work, biology, introduction to biology, we learned common characteristics of living organisms. And if I may highlight them here, characteristics of living. Okay, let me highlight those characteristics of the living organisms are let me use this characteristics of these are the characteristics of the living organisms. Living organisms, they reproduce, that is reproduction. There is respiration. There is nutrition. There is irritability. There is movement. And also there is gaseous exchange. So these are the common characteristics that we learned in Form 1 in the first topic, introduction to biology. If you can see here, all of these common characteristics in living organisms, they comprise major topics that we learn in high school syllabus. For instance, reproduction is in Form 3, we learn in Form 3, respiration in Form 2, nutrition which we learn in Form 1, irritability we learn it in Form 4, movement in Form 4, and also gaseous exchange in Form 2. So the definition of nutrition is the process. Nutrition is the process by which living organisms acquire or obtain and assimilate or utilize food the process by which living organisms acquire or obtain and assimilate or utilize food or the nutrients and this is the definition of nutrition. There are two modes of nutrition. One, modes of nutrition. The first type of nutrition is, allow me to use the other side. The first type of nutrition is heterotrophism. So this is the first type of nutrition called heterotrophism. In this type, this is where living organisms feed on. This is where living organisms feed on already manufactured complex food materials. So in this mode of nutrition, this is where living organisms feed on already manufactured complex food materials. And the organisms that exhibit this mode of nutrition are called heterotrophs. So these are the organisms that exhibit this mode of nutrition. Types of heterotrophs are, they can be I've said organisms that exhibit this mode of nutrition, which is heterotrophism, they are called heterotrophs. These organisms can be herbivorous, carnivorous, they can be omnivorous, they can also be parasites, and lastly they can also be saprophytes. So these are the examples of organisms that exhibit this kind of mode of nutrition. And this is the types of heterotrophism. Types of hetero. So I'm going to discuss about the types of heterotrophism. And I've said this heterotrophism is the mode of nutrition where the living organisms feed on already manufactured complex food materials. And these are the types. We have holozoic nutrition. We have parasitism. We have saprophytism. We have symbiosis. And these are the types of heterotrophism. If I may elaborate further, holozoic nutrition, this is where organisms ingest or take in, digest and assimilate complex food materials. In holozoic nutrition, as I have said, it is the type of nutrition where the organisms take in, digest and assimilate the complex food materials. While in parasitism, this is where one of the organism, that is the parasite, feed on or obtain nutrients from the tissues of another living organism, which is the host. While here saprophytism, it is the kind of nutrition where organisms obtain nutrients from dead decaying matter. While in symbiosis, this is where two organisms mutually benefit from each other. For example, in Lassobium bacteria and in leguminous plants, where the plant provide food and shelter, and while the bacteria fix the atmospheric nitrogen into nitrates. So this is all about this mode of nutrition, heterotrophism. Let me discuss the second type of nutrition called autotrophism. So the second type or mode of nutrition is called autotrophism. So this is the second type or mode of nutrition. In this mode of nutrition, this is where living organisms manufacture complex food materials. In this kind of nutrition, living organisms manufacture complex food materials using simple substances. Organisms that exhibit this mode of nutrition, they are called autotrophs. Examples of autotrophs are like the green plants. They are the green plants. We have algae. We also have some bacteria. So these are the examples of autotrophs that manufacture complex food materials using the symbol substances. If I may elaborate on through this definition of autotrophism, as I have said that the living organism manufacture complex food materials, and examples of the complex food materials that they manufacture are, we have the carbohydrates, also called the glucose. We have the proteins. 3, we have the lipids, or the fats and oils. So these are the examples of the complex food materials that living organisms manufacture. And these complex food materials, they are the end products of the process of photosynthesis. And for the simple substances they use, these are the raw materials, and they are the raw materials and the conditions necessary for the process of photosynthesis to take place. If I may, let me use this side. As I have said, these complex food materials that the living organisms manufacture, I've said they are the end products of the process of photosynthesis. And for the photosynthesis to take place, there must be raw materials. And also the conditions necessary are required. So for the raw materials that are needed here for the process of photosynthesis to take place, 1, we must have water. 2, we must have carbon dioxide. So these are the raw materials that are required during the process of photosynthesis. For the conditions necessary that are required, 1, we have chlorophyll molecules. 2, we must have sunlight. So the chlorophyll molecules and sunlight, they are the conditions necessary which are required during the process of photosynthesis. And for the chlorophyll are found at the granum. They are found at the granum of the chloroplast. If I may have a structure of the chloroplast, this is the structure of the chloroplast. this is structure of the chloroplast. This is the outer membrane. We have starch granule here. The starch granule. The aqueous matrix here, it is called as the stroma. And the granum which consists the chlorophyll molecule here. This one is the one that is called granum. In singular it is called granum, but in plural it is called grana. So this is in plural. So the chlorophyll molecules that are found here in the granum here. And the function of the chlorophyll, it is used to trap the sunlight. It is used to trap the sunlight energy, which will be used during the process of photosynthesis here. So, photosynthesis So we were discussing, I was at the second mode of nutrition, which was what? Autotrophism. Autotrophism has two types. One is photosynthesis. Let me write types of autotrophism. So the first type of autotrophism, it is photosynthesis. Photosynthesis, it is the process by which clean plants manufacture complex food materials using light energy. So photosynthesis is the process by which clean plants, mark here clean plants, manufacture complex food materials using what? Light energy. If you may be asked to differentiate between photosynthesis and autotrophism, I think you will see a difference. In autotrophism, we said that this is the process by which living organisms, they comprise, I said that they comprise living organisms But here in photosynthesis, it comprises only the green plants. And if you remember, the autotrophs, the examples of autotrophs were the green plants, there were algae, and there were some bacteria. So you will see that the green plants belong to kingdom Plantae, while the algae belong to kingdom Monera, and the bacteria belong to kingdom Monera. The same. So in photosynthesis it comprises only the green plants, while in autotrophism it comprises all of what? All of the organisms, the kingdom Plantae and also the kingdom Monera. The second type of autotrophism, it is called chemosynthesis. Chemosynthesis, this is the process by which organisms manufacture complex food materials using chemical energy. Process by which living organisms manufacture complex food materials using chemical energy. So here also the difference between photosynthesis and chemosynthesis, I think through this definition you will be able to draw that difference. In photosynthesis, this is where the green plants manufacture complex food materials using what? Light energy. From the word photosynthesis, photo means what? Light. So they manufacture food using what? Light energy. While in chemosynthesis, this is where the living organisms manufacture complex food materials using the chemical energy. From the word chemical energy. So next I want to talk about the stages of photosynthesis. Stages of photosynthesis. There are two stages of photosynthesis. One, we have the light stage. Okay. One, we have the light stage, also known as the light dependent stage. So this is the first stage of photosynthesis. It occurs at the glanum. It occurs at the glanum. Singular it is glanum, plural it is what? Glana. And this is where the chlorophyll molecules, trap light energy, trap sunlight energy that will be used, that is used to split, water molecules into hydrogen and oxygen gas. So that is used to split water molecules into hydrogen atoms and oxygen. If I may highlight the equation of this reaction, as I have said, it is what? The water molecule that are being splitted into hydrogen atoms and oxygen. And here we have the light energy. So the chlorophyll molecules, they trap the sunlight energy that is used to split the water molecules into hydrogen atoms and oxygen. If I balance the equation, it will be like this. So the hydrogen atoms produced here, it goes to the second stage, which is the dark stage. While the oxygen, which has been produced here, some of the oxygen will be released into the atmosphere, while the other oxygen, it is used in the process of respiration. And also there is production of energy. There is also production of energy in the form of ATP. There is production of energy in form of adenosine. In form of adenosine triphosphate. Into plaquette is called ATP. This is the energy that is produced during this process. And this process is called photolysis. This process is called photolysis. Let's go to the second stage of photosynthesis. And the second stage of photosynthesis is called dark stage. Another name for this second stage, it is also called the light independent stage. In this stage, it occurs at the stroma. I think the structure of the chloroplast I drawn here. I think you are able to see the stroma. This is where the second stage occurs. And the carbon dioxide combines with the hydrogen that were produced at the first stage to produce carbon dioxide combines with the hydrogen atoms that were produced during the first stage to produce simple carbohydrate and water. So the carbon dioxide combines with hydrogen atoms to form the simple carbohydrate, which is what? This is glucose and water. If I may balance the equation, here we have 4. So that this is what happens during the second stage. The carbon dioxide combines with the hydrogen atoms in presence of adenosine triphosphate NH to form the simple carbohydrate or the glucose and what and water. And this process is called carbon dioxide fixation. So this is the process that occurs here. And the energy used here, I've said it is called ATB, the adenosine triphosphate. And that's all about the dark stage. So next, I would like to have a discussion about the
15 minutes remaining.
Next, I would like to have a discussion on the external parts of a leaf and also the internal structure of a leaf. External parts of a leaf. Since we have plants belonging to monocotyledonous class and dicotyledonous class, so I will have, if I may have a structure of monocot leaf, I have a structure of a monocot leaf here, and also I have a dicot leaf here. So for the structure of the monocot leaf looks like this. And the external structure of a dicot leaf looks like this. So this is the leaf This is the leaf apex. This is the lamina. This is the network veins. The network veins. This is the midlip. And the last one, this is the leaf petiole. And for the structure of the monocot leaf, I have not drawn it clear, but you will be able to see. This is the leaf apex. This is the this is the parallel veins. This is the narrow lumen, the narrow lamina, I mean. It doesn't have the midlip. And this is what? The leaf sheet. So using the dicot leaf and monocot leaf, I think we'll be able to draw the differences between a monocot leaf and a dicot leaf. For the dicot leaf here, it has the midlip, while the monocot leaf, it doesn't have the midlip. Also here, in monocot leaf, we have the leaf sheet, while in the dicot leaf, we have what? The leaf petiole. Here we have the parallel veins, and here we have the network veins. The leaf is the same all through the leaves. For the internal structure of a leaf, I will not be able to draw it, but you will go back to your books and you see the internal structure of a leaf. It has the cuticle. Let me elaborate here. It has the cuticle. It has the epidermis. That is the upper and the lower epidermis. We have the palisade. The palisade layer. Okay. We have the spongy mesophyll. We have also the, and as we have the guard, we have the guard cells. So you go back to your books and see the internal structure of a leaf. If you will learn the external parts of a leaf and also the internal parts of a leaf, I think you will be able to answer this kind of a question, which is being asked as, you write the adaptations of a leaf. Adaptations of a leaf through photosynthesis. So learning about the external parts of a leaf and the internal structure of a leaf, I think you will be able to answer this kind of a question, which is being asked as the adaptation of a leaf to adaptations of a leaf to photosynthesis. It cannot be necessarily a leaf. You can even be asking, like, you discuss the adaptation of something else to its function. So for you to score these kind of questions, you need to identify these three main parts here. You need to mention the parts. You need to say how it is modified to do which kind of a function. For instance, you will say, for the one we have discussed about the external structure of a leaf, you will say it has what? It has broad lamina, 4 maximum observation of light that is used during the process of photosynthesis. I've said it as the plot lamina. Lamina is the part. How is it modified? It is what? Plot. You can say it is plot or flat. Forward function. True for the maximum absorption of what? Light that is used during the process of photosynthesis. You cannot say the pulsate cells. The pulsate layer, the pulsate cells. This is the layer that consists the numerous chloroplasts. So you will say that it has the pulsate layer which contain the numerous chloroplasts that are used during the process of what? Photosynthesis. So that's how you will answer this kind of a question for you to score the marks. And this layer, the pulsate layer, the spongy mesovilli and the guard cells, they are the photosynthetic. The photosynthetic cells. The reason being photosynthesis cell is that they contain the chloroplasts. And lastly, I will so through the guide I have been given you, you will go and write several adaptations of a leaf through photosynthesis. Lastly, I would like to tackle this part to test places of starch in a leaf. To test for the presence of starch in where? In a leaf. First, you need to expose the leaf that you want to test to sunlight at least 5 hours. So you need to expose the leaf that you need to test for the presence of starch to sunlight at least what? At least 5 hours. The next step is what? Boil water in a beaker. You need to boil water in a beaker. Then the next step is dip the leaf into the boiling water. Dip the leaf into the boiling water. Okay, in the boiling water. The fourth procedure is the reason why you are dipping your leaf here in the boiling water is to kill the protoplasm. All is to kill the leaf cells. The next step is to you put methylated spirit into a test tube. Put the leaf in a methylated spirit in a test tube. And next, stand that test tube in a boiling water or in a hot water bath for about 10 minutes. For the reason of putting your leaf in a methylated spirit, it is to It is to kill the chlorophyll or to decolorize. And the reason why we are eating the methylated spirit in dialects, it is because the methylated spirit is highly flammable. That's why we are sending that test tube in where? In the boiling picker, in the boiling water in a picker, or in the boiling water in what? In a hot water bath. And lastly, the last procedure is what? Is to wash your leaf in a picker containing warm water. The reason why we are washing your leaf in a picker containing warm water is to soften. It is to soften that leaf. And lastly, spread your leaf in a petri dish and add drops of iodine solution. Put your leaf in a petri dish, and add drops of iodine solution. So that this is the procedure of dusting the starch in a places of a leaf. And for here, using this last procedure, the color of iodine will turn to blue black, indicating that starch was present. Here we have the positive result since we exposed our leaf to sunlight for at least what? For at least 5 hours. So the photosynthesis took what? Place. So at the end we will get the positive result, which is what? The color of the iodine will turn to blue black, indicating that the starch is present. But for the negative results, you will get that at the end here, when you add the drops of iodine, the color of iodine will remain brown, indicating that starch was absent. So I would like to stop there. Thank you very much for being my audience. I'm so grateful. See you next time.